next presenter is Dr. Jane Burns. She's the director of the Kawasaki Disease Clinic at Rady Children's Hospital in San Diego and the Kawasaki Disease Research Center at the University of California at San Diego. She's known internationally for her research on Kawasaki disease, and her passion has spanned almost 3 decades with her first publication in 1982. But today she wants to talk about the KidCare study. So Dr. Burns, please.
Thank you, Guillermo. I hope everyone can hear me. We're switching back to Kawasaki disease and the happy story that the KidCare trial is now completed. So this is the trial that was initiated 4 years ago and has now finished enrollment. Thank you to everybody who's participating in this webinar who enrolled patients. We couldn't have done this without you. This was a team effort. We completed enrollment. The statisticians targeted 106 patients to meet our statistical requirements. And we were able to enroll 105 patients, of whom 100 will be evaluable for the primary outcome measure. This was a randomized 30-site comparative effectiveness trial of what you should do when your patient has recurrent fever after their initial IVIG. So it compared and randomized children to either receive infliximab 10 milligrams per kilo or a second IVIG. It was funded by the Patient-Centered Outcomes Research Institute. So the study design looked like this. Patients were admitted to the hospital and treated per protocol at the institution, but everybody who went in received IVIG. And if they had fever 36 hours to 7 days after the end of their infusion, they qualified for study enrollment. They randomized to either IVIG or infliximab. If they responded, that was fine, and they went off study after the 2-week visit. If they had persistent fever, either way, they crossed over and received the other treatment, and again, went off study at the 2-week visit. Our primary outcome measure was fever at 24 hours after initiation of study treatment. And we had a number of secondary outcome measures. There was no core echo lab, unfortunately, for this study. So we relied on center echo readings. The secondary outcome measures included, of course, safety, so adverse events, and those were adjudicated by an adverse events committee. So thank you to Ray Young, Mimi Clodet, and Margie Keller, if you happen to be on this call. The change in lab values, change in echo z-scores between baseline and subsequent time points, our secondary outcome measures, total fever days, length of hospital stay, and then patient-reported outcomes. This study was funded by PCORI, and they're very interested in the stakeholders. And so we have really detailed data. The parents were amazing. We really got questionnaire data for the first time, I think, in any Kawasaki disease study about how the parents felt their children were responding to therapy, and then observations for the 2-week period before they came back to clinic. So all of those data will be analyzed by Kathy Kim, who is the lead on that. And with that, I will turn it over in case there are any questions.
Thank you so much, Dr. Burns. We are very good on time. We will leave the questions for after the last presentation. But please, I want to thank all the questions that I already have, some of them clinical, but any research, a specific research question for our presenters, please use this Q&A box. So our last presentation for this session is by Dr. Michael Portman. He's the director of research in the Division of Cardiology at Seattle Children's Hospital and a professor in the Department of Pediatrics at the University of Washington School of Medicine. He's an attending physician at the Seattle Children Heart Center, as well as the cardiology clinic at Providence Everett Medical Center. He has been on staff at the Seattle Children's Hospital since 1992, and today he will be discussing with us the whole genome sequencing. Dr. Portman. We can't hear you yet.
Should be able to hear me now?
Yes.
Okay. So I'll launch forward. We've talked a lot about whole genome sequencing, and I'm going to focus on what we're doing in Kawasaki disease. I have no conflicts to disclose. The challenges for doing genetic research in Are you looking at my challenges slide?
Yes, we are.
Okay, good. The challenges for doing genomic research in Kawasaki relate to the marked racial heterogeneity that we all know, with different incidences in Kawasaki disease, Asian, white, Black, and Hispanic, all different. Therefore, it's difficult to develop a prognostic marker based on whole genome sequencing. The primary focus of our program has been more pharmacogenetic than looking at susceptibility issues related to genomic data. The early on the background, we have done genetic studies and focused on the Fc gamma receptor genes, which are very important as they play a role as receptors for IVIG. And there's two classifications. There's the inhibitory receptors, which are Fc gamma R2b, and as well as the activating receptors, including 2a and 3b, which are the predominant ones. You can see that the genes, which are primarily on chromosome 1, they're all aligned and some overlapping, some alternate sequencing. So it's a very complex area to do even PCR on. So we have launched to, in the past, do pyrosequencing, which is fairly detailed. The IVIG appears to restore the balance between the inflammatory activation and inhibition by the Fc gamma receptors, and the efficacy can be determined by the genomic profile. We believe that early identification of poor responders can enhance clinical trials and eventually intensify strategy. We use fairly strict phenotyping, which is really important for doing these whole genome sequencing studies. Right now, we have a biorepository of over 800 Kawasaki patients, constantly growing. We also have approximately 500 to 600 trios where we got both parents, and in some cases, we only have one parent. Within this biorepository, we will only sequence patients who have received IVIG treatment within the 10-day period, because the clinical trials have shown that that's when IVIG is most effective. And we exclude any patients who are not strictly treated by American Heart Association guidelines or where the response to IVIG is not known. With respect to, we'll go back to some of the earlier studies that we did, Fc gamma R2b. It has two promoter variants at -120 and -386 that alter transcription factor binding. And remember, 2b is an inhibitory receptor. The initial studies we did as case control. I'm sorry, my slides, there we go. As case control with genotyping, as I mentioned, performed by pyrosequencing, and race and ethnicity genetically determined not by family history or verbal, but by principal component analysis using 162 ancestry informative markers. So in this slide, you can see the racial difference based on principal component analysis, and green is the principal component analysis for Asian. Red is Hispanic, which looks separate from the other ethnic categories, but is spread over a fairly wide component, and the blue is Caucasian, which is more homogeneous. And the violet color is African American, so there's quite a bit of admixture there, which affects the racial admixture. Here you can see the extreme racial variability with specific SNPs and the frequency of the minor alleles. So you can see that there's a substantial difference between the various ethnicities in the frequency of those minor frequencies. So when we looked at Fc gamma R2b, we could see that there actually was a significant difference between responders and non-responders in genotype frequency, with the TT showing somewhat of a protective measure. So 2B we felt was fairly important and had a biological mechanism in IVIG response, and that major differences in the allele frequency between the ethnic groups could be a confounder in any type of analysis, emphasizing importance of doing accounting for race. So after we looked at the inhibitory receptors, we looked at the role of the activating Fc gamma receptors, and also found that there was a significant effect both on susceptibility and treatment response by those polymorphisms. Here again, you can see the great difference in frequency of these alleles according to race with the activating receptors. Here we show both 2A and 3B. Another strategy to account for some of these racial differences is transmission disequilibrium test. This is a family-based type test and reduces errors due to racial heterogeneity. The problem with that is that you can only use trios with heterozygous parents. We did find that there was a significant difference in the susceptibility to Kawasaki disease based on that Fc gamma R2a allele. Now, as I said, we did pyrosequencing, and this was a hypothesis-driven study, but at the same time, there were genome-wide association studies, which also identified Fc gamma R2a as an important susceptibility locus. So we also use this strategy to look at treatment response using the FBAT, as I described before. And in that case, we found that Fc gamma 3b polymorphism also was very significantly related to IVIG treatment response. In this case, looking at fever recurrence after receiving IVIG. And then secondarily, we also looked at patients who still developed coronary artery aneurysms after receiving IVIG, and found a significant effect, which was determined, which was influenced by those polymorphisms. This in particular seemed to be apparent in the Hispanic population. One of the problems, I said, with the Fc gamma region is it's overly complex. hypothesis-driven studies are often narrow. The genome-wide association studies, they contain only the most common polymorphisms, pretty much for everything across the genome. And there's already been importance shown for non-coding regions, particularly promoter sequences, that may not be picked up by genome-wide association chips or by whole exome sequencing, emphasizing the need for whole genome sequencing. Our study was proposed and funded about 1.5 years ago by the NHLBI. So we have this R01 where we can specific aims to identify genetic variations which predict IVIG resistance, identify genetic variations which can predict coronary artery outcomes. One of my major collaborators and a co-PI in the grant is Dr. Sudeep Shrestha from University of Alabama, Birmingham. They have a very large population of African Americans with Kawasaki disease. So we're doing specific evaluation in African Americans, which have not been done to this extent before. And we're also planning racial admixture mapping and analyses. Again, this is not chip-based. We're using very strict criteria to be included in the analysis. And we already have the existing DNA repository. It's sitting there and we've begun sequencing. We're using a case control method, but we're going to account for ethnicity. We also received an amazing cost from a vendor of only $600 per sample. They're offering $400 for COVID-related research. And we've already started to do sequencing. We did a first pass for quality with 12 IVIG responders and 11 non-responders. With that, we found over 3.5 million SNPs in all individuals. Most of them were annotated in the 1,000 Genomes Project database with over almost 22,000 novel SNPs found, which we're going to analyze. In this first pass with just this very small population, we found 7 SNPs of particularly interest on chromosomes 2, 4, 8, 12, and 16 that had very, very low p-values. And I have a Manhattan plot. So here's a Manhattan plot that you can see the frequency and the p-values of some of these. The ones that are above 4 are all of particular interest. I'm not ready to say which particular genes we're looking at, but I will tell you that at this first pass, we do find some genes that have some biological relevance to Kawasaki disease and may be able to explain some abnormal laboratory values that we do see with Kawasaki disease. So I will conclude there and again give credit to my collaborator, Dr. Shrestha.
Thank you so much, Dr. Portman. So I think we have a couple of minutes for questions. The first 2 questions that we have here in the Q&A box is more clinical questions, more than related to the research, but probably we can answer them. So especially for Dr. Neuberger and Dr. Randolph, do you still see MIS-C patients, or this was the only after the first wave of COVID-19? And maybe you can also address if this can change your recruitment in your research projects.
I guess I can go first. At Boston Children's, we had our huge wave in the spring, in April, May. And so now we see fewer, but what's happening in our area now is there may be a little resurgence as people have come back from college. So we may have a second wave in some communities. Other parts of the country are in the middle of their waves, and I'll let Adrian address that. So we do expect, unfortunately, a second surge. Adrian?
Yes, I have 73 centers in about 27 states, and it basically follows wherever the COVID goes. And in parts of the US, people are not into wearing masks, and the COVID activity is still very high. And wherever the COVID activity is high, the cases of MIS-C are still high. So, so we still are getting cases of MIS-C.
Yeah, in Chile, I think we saw the same thing. We have most of the cases during July, and since August, the rate was very, very low, but we expect probably to see more cases. I don't know if Dr. Burns, do you experience the same thing?
Right, so we're running a 30-site pivot from KidCare. So our same sites that were involved, just like Adrian was able to use the Polisi study to very nimbly pivot towards KD, which was great. We also were able to take our KidCare group, and PCORI went on and give us a supplement. So we're in touch with 30 sites around the United States, and it's just what Adrian and James said. There are places, for example, in Fresno, California, which is in our agricultural heartland. The Latino population there has been very heavily impacted by COVID, and now the children are being heavily impacted by MIS-C. So it's moving around. The meat packing plants in South Dakota, one of our sites there, is having a lot of COVID cases. So it's moving around.
Sure. And for Dr. Randolph, another clinical question, but maybe you can also answer how you will address this question in your research project. Which one is the best choice for vasoactive agents in MIS-C presenting with shock? And how you will address that in your study?
So, so our study isn't really designed for that specifically. We capture data, you know, of whatever clinically people are going to do. Typically, people have been using the septic shock protocols for these, but then once cardiology gets involved, they, you know, so the intensivists use epinephrine, norepinephrine, but then once cardiology becomes involved, often patients also get milrinone and other things after they see echocardiography and see if there's abnormal function of the heart. So I think that, and these are sort of the standard protocols, depending on whether you see functional impairment of the heart, whether you add some form of, you know, additional agents. But still, epinephrine and norepinephrine are the most common. People don't use phenylephrine very much in pediatric critical care, but occasionally they will have that on as well.
Great. I also have a question for the very long-term outcomes. If any of you are trying or considering to prolong the study or the research and to use some vascular function assessment to see if the MIS-C can affect the long-term cardiovascular outcomes in adulthood.
So the music study does go long-term, although for people who are completely normal always with respect to their hearts, we don't mandate any testing beyond the 6-week visit. We don't have vascular function in there, but the study is really meant for individuals to put modules on top of. So for any other organ system or type of testing, it's very available for ancillary studies.
Could I? I have a question to the panel. Everybody is worried about the MIS-C in terms of shock. How easy is it on a clinical grounds to differentiate what is vascular oozing, microangiopathy, and myocardial injury or pathophysiology in these cases?
I think that so we're doing a study right now to identify these subphenotypes in MISC, and looking at a lot of different criteria, including all the symptoms people come up with, because we have 2,000, patients in the data set and 400 with MISC. And then some patients are very inflamed, have elevated BNP, have all the features, but don't have 2 organ systems involved. So that it's such a spectrum. And I'm very worried that these criteria we came up with, or, the CDC criteria and the other criteria from other countries is overlaps with so many things, including acute COVID-19 with shock and ARDS. And so it's just, the criteria are very problematic. And so, just not to, I didn't really answer your question, but I think that these are, this is the biggest challenge right now. What are the markers and what are the criteria?
I think it's a good moment to stop right there. We are almost on time, so thank you again for the speakers, for these awesome research projects, and I hope they illuminate all of us to improve our management of this disease. Thank you so much.
Great. Thank you, everyone. Jane, Adrienne, Jane, and Michael. Also, thank you, Robert. Thank you for sharing your knowledge and for sharing your trade secrets. We're either within the IKDR or on separate consortia. I think it is very valuable to patients all across the globe. And this is also the goal of the IKDR, to be as inclusive as possible, including very prominent research groups from our presenters today. I'm very amazed by today's webinar. I'm very happy that this is one of the first webinars where I would have the ability to wear trousers rather than boxers or PJs. I hope everybody was comfortable at home. I would like to give everybody a virtual hug and also ask Brian to take the microphone, address the audience. But prior to that, I would like to thank particularly Sunita, who had been gluing the IKDR together by perfect messaging, taking answers, taking requests, as well as everybody else who helped in putting up this whole day of a meeting. Brian?
So I, first of all, want to thank actually Najib and the team in Montreal for hosting this virtual meeting, although we're not in Montreal. And so we're not able to partake of Najib's excellent hosting skills like we have in the past. But here's hoping that in 2021 that we all can get together actually in person. We should have lots of stuff to share by then. I'd like to thank all the centers that have joined the IKDR for their interest. Hopefully we'll get everybody up and onboarded and the data collected soon. And I want to thank especially all of our speakers today. It was really an excellent forum, and we're going to be keeping this up ongoing in the future. So I think we're already planning our next open webinar, which is going to be on the assessment of coronary arteries. We have yet to set the date, but we'll certainly be in touch with everybody. So I want to thank everybody who participated, and thanks to Najib and thanks to Sunita for organizing this great meeting. Thanks.
Kampai.
Kampai, yeah, kampai.
See you next time in Montreal live.